The overall goal of this procedure is to visualize the location of viral RNA in situ within cells. To accomplish this, first grow the cells that will be transfected or infected on glass cover slips. The second step is to fix the cells with 4% paraformaldehyde after successful transfection or infection. Next, incubate the cells with a hybridization mixture that labels the RNA. This is done by placing the cover slip cell side down on the hybridization mixture on a slide. The final step in this procedure is to use antibodies to stain the cells for the RNA label. Ultimately, the localization of the labeled RNA within the cell is visualized by fluorescence in situ hybridization, or FISH.
The main advantage of this technique over other methods, like RNA imaging in live cells using tags, is that native RNA can be detected, as opposed to modified RNA.
Generally, individuals new to this technique will struggle because it requires patience and care in handling cover slips, standardization of method, as well as precise timing during incubations.
The specific procedure for culturing cells for fixation will depend on the type of cells. For adherent cells, it is initially important to seed them onto untreated, sterile glass cover slips, so the final confluency upon harvesting is approximately 70 to 80%. Non-adherent cells should be cultured the standard way until they are ready to be collected. Then incubate them with cover slips that have been treated with poly-L-lysine. For a typical 12-well polystyrene tissue culture plate 150,000 cells per well at 24 hours prior to infection or transfection. Incubate the cells on the cover slips for 1 hour at 37 degrees Celsius. To begin the procedure for cell fixation, discard the medium.